Good morning, everyone. My name is Abdul Malik Setiawan. I am from Anatomy Department, Faculty of Medicine, University Kebangsaan Malaysia. Today, I would like to talk about my study on BMP4 expression enhance the capacity of human adipose-derived stem cells for corneal differentiation. This is my outline for today's presentation. Okay, corneal blindness is an eye disease that characterized by the permanent opacity of the cornea. Trauma, infection, and autoimmune disease that happen in the transparent layer of cornea and it is untreated can cause permanent corneal opacity. The gold standard therapy for this disease is by corneal transplantation. However, limited number of donated cornea make this disease become a major health problem. So what is the alternative? Tissue-engineered corneal epithelial cells derived from stem cells could solve this problem. Adipose-derived stem cells is one of stem cells that arise from mesodermal lineage. Has multipotent capacity and also it's abundant and easy to isolate. ADSC also has less ethical concern for its procurement. To know the stemness and pluripotency of the stem cells, people always use transcriptional factor level, such as octamer binding transcription factor. Octamer 4 maintain stem cells pluripotency by suppressing the stem cells differentiation. Recent studies show that bone morphogenetic protein 4 is suggested to induce stem cells differentiation into ectodermal lineage via TGF signaling pathway. Few studies already been done to differentiate MSC into corneal epithelial cells. Several methods such as chemical induction, co-culture, conditioned medium, and 3D scaffold system were already been deployed. Most of the study used cytokeratin 3 and ABCG2 as a marker for successful corneal differentiation. Sikora in 2019 showed an increase of CK3 expression after they co-culturing the ADAC with porcine limbal epithelial stem cells. But no one has done to compare the capacity of subcutaneous and visceral ADAC. So in this study, we evaluate the basal BMP4 in primary human ADAC isolated from subcutaneous and visceral adipose tissue depot. And we want to know the correlation with the differentiation capacity toward corneal differentiation following co-culture method. Our study consists of two phases. In the first phase, we do ADAC isolation and we measure basal BMP4 and octamer 4 evaluation. After we isolate the fat from 6 consented women, we culture the ADAC until passage 4, and we did MSC characterization by flow cytometry and multi-lineage differentiation. In the second phase, we culture both ADAC, subcutaneous and visceral, using co-culture method with human telomerase corneal epithelial cells. We use control, which is undifferentiated, and after 15 days of differentiation, we do immunofluorescence staining and real-time PCR to know the expression of protein and gene of ABCG2, octamer 4, BMP4, and CK3. The tissue collection was done in 2022 and 2023. We've collected from 6 productive healthy donors. The average size of the sample was 7 grams. So in the first phase of the result, we did the MSC characterization. All the subcutaneous and visceral ADAC were adheres to the plastic. All were able to differentiate into adipogenic and osteogenic cells following induction. and all express CD73, CD90, and CD105. However, the level of the MSC marker were lower compared to the ADAC cell line. And the visceral ADAC also showed lower MSC marker compared to the subcutaneous one. So in the octomorph4 and BMP4 basal gene expression, we showed that BMP4 in visceral ADAC was significantly higher in visceral ADAC compared to the subcutaneous one. And the visceral ADAC also expressed higher octomorph4 compared to the most of the subcutaneous ADAC. So following 15 days of co-culture differentiation, we know that the ABCG2 expression in visceral ADAC was decreased in comparison to the subcutaneous one. In the contrary, the visceral ADAC showed an increase in CK3 expression following 15 days of co-culture differentiation. For immunostaining, we saw that both subcutaneous and visceral ADAC has an increase in CKT protein expression. However, in visceral ADAC, the difference is significant. On the contrary, for subcutaneous ADAC, showed an increase in ABCG2 protein expression, and it's significant. But the visceral ADAC has decreased in ABCG2 expression. So from this study, we know that both subcutaneous and visceral ADAC has different basal BMP and octomorph4 level. The visceral has higher BMP4 and octomorph expression. And after 15 days of co-culture differentiation, the subcutaneous ADAC increase in ABCG2, while visceral ADAC has higher CK3 expression. So this is in line with study by Ritter in 2019, where he showed that visceral ADAC has higher stemness-associated gene other than octomorph4. And in 2020, Kobayashi showed that BMP4 initiated ocular surface ectodermal formation via TGF-beta signaling pathway. So that's why in visceral ADAC has higher differentiation into CK3. And maybe other pathway also lead to corneal epithelial differentiation, such as SHH pathway. So in conclusion, higher corneal differentiation capacity in visceral ADAC might be due to its basal BMP4 expression. And for future study, we suggest to our other researcher to pay more attention in visceral ADAC. And we also suggest to do longer co-culture duration and also incorporated other differentiation methods such as chemical, secretome, and 3D system. I would like to acknowledge a C term for ADAC cell line procurement, obstetric and gynecology department of Hospital Cancer Tuan Kumuhris for samples collection, and other fellow science and research officer in anatomy department, and other fellow postgrad student. I also want to thank to Faculty of Medicine UKM for the funding this research. And this is my reference. Thank you.